Welcome to The Explainer. Today, we're diving into a scientific paper that honestly reads more like a medical detective story. We've got a routine experiment, an invisible enemy, and a sudden, desperate race against time. Let's get right into it. So our story kicks off in a research facility at Western Sydney University, and our main characters, they're a colony of spinifex hopping mice. These are small native Australian rodents, and they're famous for their really unique two-legged hop. They're pretty common in research, and in this case, they were the subjects of what was supposed to be a very, very simple study. Okay, so picture this. 11 of these little hopping mice are selected for a simple experiment. The whole point was just to see how they do on a diet of nothing but sweet potato. You know, it's a pretty standard type of study to figure out an animal's dietary needs and how flexible they can be. And for the first 5 days, everything was completely normal. The mice were eating, the researchers were taking notes, everything was going exactly to plan. There was absolutely no reason for anyone to think that a deadly outbreak was silently brewing right under their noses. And then, bam! Everything changed. What started as just a routine nutrition trial suddenly spiraled into a full-blown crisis. The researchers had to drop their clipboards and basically become first responders. The whole situation just fell apart with terrifying speed. On day 6, one mouse gets sick. By day 7, it's dead. A few days later, another one dies. And then on day 11, it was an explosion. 4 more mice were found dead. And get this, some of them weren't even in the trial. That was the moment everyone knew this wasn't just a bug. This was something much, much worse. In the end, a total of 11 mice were lost. This wasn't some minor sickness. It was a catastrophic event that put the entire facility at risk. They had to stop everything and figure out what in the world they were dealing with. So with the death toll climbing, the team just slammed the brakes on the whole experiment and launched a massive investigation. They weren't just scientists anymore. They were detectives, hunting for a killer they couldn't even see. Their first big break came during the post-mortem exams. As they were looking at the deceased animals, they found a huge clue: tiny cream-colored spots all over the surface of the livers. These spots, called foci, are basically little areas of tissue damage. This was a vital piece of the puzzle. This discovery really narrowed the search. That specific kind of liver damage pointed toward a certain type of infection. So samples were rushed to the lab to be put under a microscope, all to identify the culprit behind this incredibly fast and silent killer. And when the lab results came back, the invisible enemy finally had a name. And it was an infamous one, a pathogen known for striking with almost no warning. The diagnosis was Tyzer's disease. It's caused by a nasty little bacterium called Clostridium piliformes, and they confirmed it using this special silver stain that, under the microscope, literally lit up the little rod-shaped bacteria hiding in the damaged liver tissue. And here's the thing about Tyzer's. It isn't just a mouse problem. Not by a long shot. It's a known threat to a huge range of animals. We're talking everything from pets, like rabbits and hamsters, to horses, and even wildlife like koalas and red pandas. So understanding this disease is a really big deal for vets and conservationists. But what makes Tyzzer so incredibly dangerous is that it's often a completely silent killer. This quote from the paper says it all. Animals can seem perfectly healthy 1 minute, and then they're just gone. That lack of warning signs makes it almost impossible to diagnose and treat before it's too late. Okay, so they'd identified the killer. That's step 1. But now came the really hard part. How do you stop a highly infectious, microscopic enemy before it spreads even further? See, the real weapon this bacteria has is its ability to form spores. You can think of them like tiny, armored seeds of disease. Unlike regular bacteria, these spores are super tough. They're resistant to most disinfectants, and they can survive in the environment for up to a year, just waiting for a new animal to infect. So, to fight back, the facility went into full-on lockdown mode. They switched to metal bowls because they're way easier to sterilize. They made sure to care for any sick animals last to avoid cross-contamination. But the real game changer was bringing in the heavy-duty stuff, a powerful disinfectant, basically a strong bleach solution, which is one of the only things that can actually kill the spores. And this led to the most gut-wrenching decision of the entire crisis. 2 mice had actually survived the outbreak and seemed perfectly healthy. The problem was they could be asymptomatic carriers, silently shedding those invisible spores and putting every other animal colony at risk. It was a heartbreaking choice, but to make sure the disease was truly gone, they had to be euthanized. So the outbreak was finally over, but what it left behind was a whole set of critical, hard-won lessons for pretty much anyone who cares for animals, from research labs to wildlife sanctuaries, and yeah, even pet owners. Looking back, the researchers figured that stress was the most likely trigger. Things like a sudden diet change, or even just being housed alone, can weaken an animal's immune system. That's all the opening and opportunistic bacteria like this one needs to take hold and wreak havoc. Ultimately, when a disease moves this fast and hits this hard, treatment just isn't realistic. Prevention is everything. The biggest takeaways are you have to minimize stress on animals, you have to use the right kind of spore-killing disinfectant, and you have to be ready to quarantine at a moment's notice, because as we saw, even an animal that looks healthy can be a hidden threat. You know, this whole story about the hopping mice started from a simple nutrition study, but it serves as this powerful and frankly sobering reminder of a much, much bigger challenge. It really forces us to ask, how do we get better at protecting the vulnerable from the invisible threats that are all around us? And that's a question that extends far beyond the walls of any research facility.